So hello, everyone. Welcome to this seminar. This is going to be, very unique seminar in terms of how to use microscopy and super resolution microscopy, new techniques that they have been around not from so long. We have an exceptional presenter today. He is Dr. Jakub Koinaki. Dr. Koinaki has been working in different places. Actually, he did his training at Burnet Institute at the University of Melbourne in Australia, where he obtained his PhD in science. Then he traveled from Australia to Europe, and in 2007, he started a postdoctoral position at Heidelberg University in Germany, where he worked on visualization of HIV substructures using the super resolution microscopy that he's going to be telling today about. Then a few years later, in 2013, he joined the laboratory of the Witherell Institute of Molecular Medicine at the Oxford University in the UK, where his projects focus more on the mobility of lipids and proteins on the surface of individual HIV particles and at virus assembly sites. Remember that HIV is a retrovirus that is the etiological agent of AIDS. And now, since March 2018, he is part of our AIDS Research Institute, IrsiCaixa, in Badalona at the Germans Trias i Pujol Hospital. And he's working on live cells imaging studies on, again, HIV virus cell interactions, as well as establishing a biosafety level 3 super resolution imaging facility, which is especially catchy in these moments because we need this type of level 3 biosafety laboratories to work with SARS-CoV-2, the virus of COVID-19 disease. So you'll see that he will introduce you in theoretical and practical terms, and he will doing something very exceptional, which is showing you real imaging of virus, both HIV, the H viruses, and also SARS-CoV-2, the COVID-19 virus. So having said that, I will leave you with him. And by the end of the presentation, I'll be happy to coordinate any questions you might have for Jakub. So thank you, Jakub. The floor is all yours. And thank you to all the attendees for being here with us today.
Great. Thank you, Javier. Thank you very much. Thank you for the organizers for letting me also present this workshop to you. Okay, and so what I will do is, what I'm going to do is first I'm going to share the screen, my screen here, and I'm going to first talk a little bit about some theoretical information, theoretical background of how do microscopes work. Just to give you an idea of later on when we move to the practical demonstration, how is everything working. So let me share the screen and start the presentation first. Okay. Hopefully that should work. Hopefully you see what I'm saying. So today I'm going to talk to you about how viruses, how to image viruses as a scientist. Obviously, it's a very important time, and also microscopy is a very interesting, a very important tool. So let me get started. And what I'm going to do is, first of all, in terms of what I'm going to do in the first part of the presentation, I'm going to give you a bit of an overview on virus size and architecture, and I'm going to then talk a little bit about the principles of microscopy. Then we're going to go for a bit of a basic anatomy of the microscope. Actually, I hope you see a little my camera as well as the screen that I'm sharing, because I will, in addition to showing some things on the screen, I'll also be wanting to point out a few things on the as I talk to you about the microscopes. I want to show those things to you via camera. Then we're going to talk a little bit about introduction of what's so-called super resolution microscopy, of course, providing all the background what resolution is and what is this thing called super resolution. And finally, I will touch upon how this super resolution microscopy is very useful for studying virus particles. And finally, I have a demonstration prepared for you where what I'll do is then I'll switch off the presentation and I'll switch the screen sharing to the actual software for which I acquire the microscopy images. So you'll be able to, first of all, via camera, see me operating the microscope, but also on the screen shared, you'll be able to see what I see as I acquire the images, microscopy images, and I'll obviously explain everything as I go along. And then at the end, I'm gonna say a few words on actually what did we see, and provide a bit of outlook on the results of today's workshop. So the first concept that I want to introduce you to is, well, the size. Viruses are really, really small, and we're talking about nanometers small. But for us humans, it's really kind of difficult to appreciate just how small that is. It's actually very difficult to show it on the screen. So I mean, this is a graphics that I found that tries to a little bit to just give you an idea how small they are. So on the right side, you have a human hair, which of course is very thin, and you can see in comparison how small the viruses are. So obviously they are invisible to the naked eye, and we need powerful tools to study them, study the structure, or how do they interact with human body. And just to give you an idea, just exactly how small those viruses are. And there are, of course, different types of viruses, and they have some variance in size. But the viruses that I'll be talking about today and we'll be imaging today, first of all, we have HIV. And on the screen, you can see a structure of HIV. And that virus is 140 nanometers in size. And what does that mean? We're talking about 140 times 10 to the -9 meters. So it's very, very, very small. We talk all the viruses, we're talking in nanometers. Another example of the virus, which actually does not look like a HIV, because HIV is very spherical, is a well-known Ebola virus. And this virus actually has a filamentous structure, but its diameter is also about 80 nanometers in size. However, it's very long, it can be very long and thin, and we're talking about up to 800 nanometers, which is 0.8 of a micrometer in size. And then, of course, we have, very famous right now, unfortunately, SARS-CoV-2 virus. And that virus is about 100 nanometers in diameter. And that makes it just a little bit smaller than HIV. Just a little bit of a note. I mean, I'm sure you have seen in the science books and even in a variety of informative graphics that scientists often picture viruses as as spheres or just circles, and you see some proteins. You have some surface proteins. You have internal structure proteins, and it all looks fairly actually sort of empty. However, if you actually look at the more accurate simulation of a virus, you can see how full of proteins and lipids and many components it is, genetic material, proteins. So viruses are actually extremely complex structures, which we sometimes do not appreciate when we just look at the very basic graphics. So to study them is actually a complex science to be able to realize just how do they interact with human body, with cells that they infect. It, as I was mentioning before, requires some powerful tools to visualize those very, very small things. And how do we do that? Well, one of those tools is fluorescence microscopy. And it is one of the, for me, a pillar of scientific discovery. First of all, it provides, images directly. And to us humans, seeing is believing, right? So when you observe something directly via the ocular, it makes it a little bit more real. Because a lot of science, of course, relies on indirect readouts, where you get a lot of numbers, a lot of graphs, and those are not so directly informative. Sometimes, as when actually seeing something with your own two eyes. So, as a scientific tool, it's very, very important. And so, what are some of the features of fluorescence microscopy? Well, why is it useful to scientists? Well, first of all, as you can see, actually, I'll go from the start. First of all, when studying biological systems, we often want to avoid any harsh chemical treatments. We want to study things as they are without subjecting them to strong chemicals, which may alter the structures of cells, the structure of viruses, which of course is a problem because it then does not give you exact idea of how it works in physiological conditions in a human body. So the microscopy can be performed without the need for harsh chemical treatments. Another important thing that leads from that point is that microscopy is possible for live cell imaging. So here is an image of an immune cell in a tissue, surveying the antigens and eating foreign material. So fluorescence microscopy allows you to study things live, which is very important because obviously, once again, we can study things as they are without altering them too much. Another very important thing is that we can, fluorescence microscopy allows us for specific tagging of multiple targets. And this is exactly what this picture on the top shows. With microscopy, fluorescence microscopy, we can focus on very, very specific things that we want to observe, we want to study, and do not worry about all those other things which we do not want to look at. So here you have images of the cells on the top right. The cells have been stained for one color is a nucleus, another one is a cytoskeleton in green, and in red you have I think another portion of a cytoskeleton as well as membranes. So with fluorescence microscopy, we can, once again, like I was mentioning before, we can look at things, we can visualize things only what we want to and discard all others. And a final point is, well, that compared to other techniques like electron microscopy, for example, it is, well, relatively simple and fast. It's fairly easy to prepare the samples. And as you will see, it's actually fairly easy to, with a good microscope, to study samples, to image them, and get useful data. Of course, sometimes it takes a lot of work to acquire, a lot of useful data. Sometimes the samples don't work, but it is, I would say, in my experience, it is relatively easy and fast. So these are the advantages. Now, of course, no scientific technique is without its disadvantages. And the very important disadvantage of the fluorescence microscopy, especially for study of the viruses, is its limited resolution. And remember, like I was saying, that virus is 100 nanometers in size. And remember that number, because I'll get back to it in terms of resolution of the microscopy. So unfortunately, with the limited resolution of classical standard fluorescence microscopes, viruses were normally studied using electron microscopy. So here you have an image of electron microscope as it imaged the HIV virus particles. And classically, until about early 2000s, study of the structures of the viruses was purely in the domain of electron microscopy, which has a lot of disadvantages, like I was saying, harsh chemical treatments, and no potential for live cell microscopy. But well, before we get to study of tell you about super resolution and fluorescence microscopy, I want to also give you a little bit of a background on what exactly fluorescent fluorescence is, and what is fluorescent labeling, since these are very important central aspects of understanding fluorescence microscopy. So in a nutshell, if you look on the top left, what is fluorescence? Effectively, fluorescence is when you provide an excitation photon to a molecule, to an atom, which causes its electrons to jump into a high energy state. And when those electrons drop into the lower resting energy states, they emit a fluorescent photon, photon of light, which is often of very specific wavelength. And this we can detect with the microscopes. So here is, for example, an example of a spectrum of a fluorescent molecule. So in the graph in the blue, we can see that the molecules can absorb certain photons of light of certain wavelengths. So by 400, 500, we're talking about visible light here. We're talking about blue light, and then you can see on an orange peak, these are the wavelengths of light that they can emit. And as you can see, those graphs, those peaks are not exactly the same. And why is that? This is because there is always some energy loss when we are talking about fluorescence emission. So when the electron is excited with the blue light, it tends to emit higher wavelength orange light. This is because blue light is generally more energetic, and the red light has a little bit of less energy, and that is exactly why this event called Stokes shift This is why the fluorescent molecules fluoresce always a little bit towards orange or red, part of the visible spectrum. And this is visible by this, higher wavelengths of the emitted photons compared to absorbed photons. So, what are some molecules that we use as a scientist to study fluorescence microscopy? And we use several different molecules which have exhibit this fluorescent behavior. And one of them is so-called classical green fluorescence protein. This is a green fluorescent protein which, when excited with a blue excitation light, will give you a green emission light. And actually, in the samples that I'm going to show you later today, I actually have a virus that is labeled with a green fluorescent protein. However, well, proteins are often very big, so we also have another alternative, and this is so-called organic dyes. And organic dyes, as you can see, are much smaller molecules, because this is a protein. This has a lot of, it's much bigger than a small chemical compound like this. So here we, as scientists, we also have a possibility to use a very small dyes, which we can attach to structures of interest, like proteins, or lipids, or sugars. And those molecules also will fluoresce with a specific light. But how do we attach those fluorescent molecules to the structures of interest? Well, that is normally realized by utilizing antibodies. As you may know, antibodies are part of our immune system, and they are an important part of our immune systems, as their role is to attach to very specific molecules called antigens. They recognize very specific molecules, and then when tagging, when those molecules are touched by antibodies, then our immune system can recognize those. And we have taken advantage of this, and we, scientists, have started utilizing those antibodies to be specific to structures of interest in fluorescence microscopy. So what we do generally is, when we want to stain the sample for fluorescence microscopy, we'll have antibodies which are specific to the structures that we want to study, and those antibodies also have the fluorescent molecule attached to another end. So by utilizing specificity of antibodies, we can target the structures of interest in viruses, cells, tissues, all the way to complex samples. Okay, so that is a bit of a background on fluorescence and fluorescence labeling. Now let's talk a little bit about microscopes. There are many different types of microscopes, and they often serve different purposes. Some are more useful for certain type of analysis. Other fluorescence microscopes are useful for another ones. And here is an example of the machines that I have, for example, personally worked with. And as you can see, these are often, well, very complex machines. And I have here one of them, which is a little bit more simple, but also a very powerful machine. And I will tell you about some details of it in a moment. But just to show these images for you to appreciate a little bit that these are actually quite complex, large, and well, expensive devices, which are used for fluorescence microscopy. And these are generally actually commercial systems. So these are companies build them, and then scientists use them. However, what scientists also do is, well, we actually build our own microscopes. So we can actually, if you want to have a microscope that is specific for for some very specific type of a study, or you simply want to investigate a new type of technology associated with microscopy, because, well, in addition to biology development, there is also technological development. Cameras get better, detectors get better, lasers get better. We also build our own microscope. So here I have an example from the lab that I was in, where we actually were building our own microscopes. And you have an example here of a laser light path, which actually we use to excite the molecules. Because what I forgot to actually mention is, how do we excite those fluorescence molecules? We actually use lasers with a very specific wavelength, which are corresponding to those absorption spectra of the fluorescent dye. So we have also possibility to play with to build microscopes, and it's a little bit like playing with Lego blocks. Actually, the putting all these components on the board, on a table, is a little bit like playing with Lego blocks. And actually, And there are often projects, there are also projects which actually utilize Legos to build your own microscopes. And if any of you is interested, here actually in IRB in Barcelona, on this webpage, there is an example of a project which allows you to build your own quite actually powerful and useful microscope utilizing common elements from Legos. So if you're interested in setting up your own project and building your own microscopes from Legos, there is this project, and there is a couple of others on the internet, which allow you to build quite actually powerful microscopes, so you can test all those things yourselves. Okay, so let's go into the anatomy of the light path of the microscope. How does a light, laser light or fluorescent light, travel inside a microscope? So here on the screen, I have an example of a basic microscope body. So just to give you a little bit of a perspective, generally the microscopes have this. Most of the microscope, like you maybe saw on the previous images, have a central part called microscope body, and this is quite often distinguished by having an ocular. Well, this is a little bit of I guess maybe a throwback to the old times where always microscopy was done with our human eyes. And well, while human eyes are very, very powerful tool, because they're connected to our brains, which is, of course, an extremely powerful processing, well, not a computer, but processing organ. And in many ways, it's actually very useful to still see things with your own eyes. So that's why a lot of microscopes have oculars on them. However, that is not really necessary. There's nothing stopping us from building a microscope that just has the other elements where you detect fluorescence, and you do not need this ocular. However, many of the microscopes you will see, even the commercial ones, still retain this part, because as I was saying, seeing is believing, and also being able to see the samples allow us to quickly process that information without relying on the computers, because computers can process the information they're also very powerful, but they generally sometimes are limited to the tasks that they are that have been specified to, and whereas we are a bit more versatile. So let's look at the fluorescent microscopy light path. How does a light travel in the fluorescent microscope? So first of all, we have the fluorescence light source where the light comes in. Do not worry about the bright field light source. I'm not going to talk about this. This is slightly different. A bright field microscopy is, well, it's not a fluorescence microscopy. It's more classical one, but we're going to focus a little bit, focus today on just fluorescence. So fluorescent light generally enters from the back of the microscope. And here, if you can see on the camera, actually that corresponds to here on the back, that fluorescent light comes in here. Then the light travels, is reflected by the mirror and goes towards objective. And objective is a very important part of a microscope. You cannot see it here, but objective is a very important part of a microscope where this light is focused on the sample that we put here. Afterwards, when you excite the molecules of fluorescence, then the same objective collects the fluorescence that has been detected, as in that has been the fluorescence that is being emitted by the excited molecules. And the same light, as you can see on the arrow, travels down. And then we can direct it either to the ocular, so to see it, or we can direct it to the detectors for detection via very high sensitivity fluorescent light detectors. And in this microscope, exactly is what happens. We can either ask the light to be fluorescent light to come to the ocular, to our detector, to our eyes, or then we can ask it to be sent to this element here, which contains machinery to send it to the main box of the microscope, which contains all the detectors. So actually, a little bit of additional information about this setup over here. The microscope, all the lasers and all the detectors are here, and then the fluorescent light is sent via optical fiber to this so-called scanner head, to the scanning unit of the microscope. The laser light travels to the objective, returns to the scanner, and then is sent back to the main control unit where it is detected. Okay, another concept that I need to introduce to you, for you to understand what is happening on the screen later, is what sort of type of the microscope that we have here. And the microscope that we have here in front of us is called confocal laser scanning microscope. Okay, what does that mean? First of all, confocal. What does mean confocal? Confocality means in focus. So it means that this microscope allows us to detect only the things, only the fluorescent photons that are in focus of the sample. So anything that is above and below the focal point of the laser light is discarded. So this is very important because when you often see on a microscope sample, when you don't use the confocal microscope, you see a lot of light coming from different parts of the sample, and that makes it much more difficult to observe things that are in focus. Another thing, what is laser scanning microscope? Laser scanning microscope works like this. Okay, so for example, just to give you an example, how does it work? We have, for example, two virus particles, which we have labeled. We have labeled some surface proteins, and we have the fluorescent, yellow fluorescent molecules. And we want to observe them using this microscope over here. So confocal microscope, so laser scanning microscopy, what it does is when we ask it to scan, when we ask it to look at the sample, it basically scans a laser beam of light in a line pattern, and then it reads it out. Think of it a little bit like an old TV, where the electron beam used to scan across the monitor in old TVs, and that generated you the image. This is exactly what is happening here. The laser beam is scanning very rapidly across line by line, and then also that line by line gives you a fluorescent readout. So you have excitation, and then you have the fluorescent readout, which then gets detected as a final image. And you may have already noticed here that how big this fluorescent readout is. And this comes back to the fluorescent resolution limit of the microscope. See, we labeled the ground truth. We labeled things which are very, very small, but we get a very, very, very diffuse fluorescent image out of that. And this ties in to the concept of resolution of the microscope. Remember the numbers of the numbers that I told you before about the size of the viruses. So fluorescence microscopy resolution limit states that the resolution of the light microscope is approximately 250 nanometers in XY direction. Is when we're looking at the sample in XY, and it is limited to resolution of 600 nanometers in the Z direction when we look things in and out of focus. And so, what does that mean? That means that if you want to image any object that is smaller, that is approximately 250 nanometers, the fluorescent readout that we're going to get is a fluorescent sphere of about 250 nanometers. So remember, like I was saying, viruses are about 100 nanometers in size. So if you want to study a portion of a virus or a structure within a virus using a conventional microscope, well, then anything that is smaller than 250 nanometers in size, let's say like an object, let's say, for example, here 40 nanometers, always is going to give us a fluorescent readout of 250 nanometers. So as you can imagine, we cannot really get any information about structure of the viruses that are smaller than 250 nanometers. And what is the cause of this? Well, without getting into too much details, first of all, this is due to the wave-like nature of the light. The photons interfere with one another. And also, this comes to this resolution limit is also because we cannot collect entire fluorescent light that is emitted by fluorescent molecules. So this, for many, many years, since this type of microscopes have been introduced since around the 1980s, or even a little bit earlier, but around that time, the laser scanning confocal microscope became viable and used. Since then, normally, since the 80s, you could not really use this type of microscope to study virus structures, precisely because of this resolution limit. But in around early 2000s, this has changed. And with around, late 2000s, let's say 2004, 2005, a new concept started emerging in fluorescence microscopy, and they were called super resolution microscopy techniques. And what does that mean? Effectively, very, very clever people try to come up with a way on how to, well, not so much break this resolution barrier, because the resolution barrier is down to some physical laws, but more like try to, well, cheat it a little bit. try to overcome this resolution barrier and allow fluorescence microscope to see things below these 250 nanometer limits. And this has resulted in quite a lot of new types of microscopy in early 2000. Well, they have a very complicated names. Don't worry about those. But just I wanted to tell you that it was sort of a revolution in the field of fluorescence microscopy. And it was a revolution of such size that in 2014, the inventors, the main inventors of these techniques, superposition techniques, got awarded a Nobel Prize for it. And as to showcase how important this was to scientific discovery. And me, myself as a researcher, I took advantage of these techniques, and really I can tell you that it has allowed us, especially people who study viruses, to see things which were not be able to see before using fluorescence microscopy. So one of the techniques that I will introduce to you is for superposition microscopy that I will introduce to you in briefly is Stead Microscopy. Stimulated emission depletion microscopy, because this microscope that we have here is not only a standard confocal fluorescence microscope, it is actually a super resolution microscope, which means it is not only capable of standard fluorescent confocal imaging with a normal resolution of 250 nanometers, but it's also capable of super resolution microscopy. And that is going to be very crucial for us when we still look at the viruses. What I'm going to do is I'm going to try to explain a little bit on how this microscopy works. So to do that, we have to look into what is happening when we try to image something in the microscope. So here we had an objective. We're focusing the laser light on a sample. Okay, so to walk you through it, the green thing, the green cone, is a laser light that is being focused. Two red spots here is two hypothetical objects. Let's say they are 40 nanometers apart. As you can see, when we focus the laser light, at some point in time, I mean, when we look at it, when you think about it, sort of like a common sense, you think that you can focus the light to infinity. But that is not true. The laser light can only be focused to a certain point upon which, like I was saying, the photons of light start to interfere with one another, and you have imperfect focus, where you have this sort of a diameter of the which is a limit of the focusing of the laser light. So what happens when we try to image with those 2 points, which are 40 nanometers apart, using a standard fluorescent microscope? Like I was saying, here we have the excitation volume of the laser, and let's see what happens when, we use the laser scanning microscope to scan those 2 molecules. Okay, so what you can see here is both of them light up at the same time, and the signal that you get out here on the left is, well, shows, well, that we do not see a separation. We do not see a separation between those 2 points. And this is exactly where the resolution limit of a fluorescence microscope come in. That because of the width of this laser focus spot, we cannot distinguish that there is a distance, some distance between those. And the signal that we're gonna get is indicated that there is, well, just one fluorescent spot. Now, Stead superposition microscopy overcomes this limit by introducing a second laser into the mix. And without going into too much detail, this laser has 2 features. First of all, it has a specific wavelength which allows you to inhibit fluorescence of the molecules that are present within the field of this laser. And second of all, this laser has a very interesting shape. It has the shape of a donut. You may have seen some donuts that I put on the title page. This donut has a very powerful laser intensity here in these areas, but has a 0 intensity in the middle. So let's see what happens when we try to scan those 2 hypothetical points using Stead superposition microscopy. So the laser will scan again backwards. And as you can see, actually, I'll go back a little bit here. See if I can. We start seeing that now the signal, we can start detect that there is a separation between those 2 molecules. And this is what happens here, that despite this molecule over here on the right being in excitation field, it is depleted or inhibited by a second Stead laser, and only the molecule that is present in the central 0 intensity in the middle is allowed to fluoresce. So in a very simple terms, what we simply did is we squeezed the excitation volume, the fluorescence excitation volume, from this quite large 250 nanometer diameter size to something that is much, much smaller. And this microscope is able to give you a 20 to 40 nanometer resolution, which is a very, very, very big improvement. We're talking about 10-fold improvement into the resolution. And the 20 nanometers is much, much smaller than a virus size, which becomes important. Okay, let's go to the next part. Okay, so finally, let's try to tie it all together. Okay, so we have a powerful tools for study. We have viruses. We now can study these viruses in superposition microscopy. And all I want to tell you is that as scientists over the last 10 years, we've been taking advantage of these techniques. And HIV is actually incredibly has been one of the very first viruses that have been used in conjunction with superposition microscopy. So we can study things like assembly and release. We can study virus architecture, virus entry, and post-entry stages. Here you have just an example from the scientific paper showing how these techniques have already been used to study all sorts of different parts of the virus replication cycle. When viruses come out from the cells, when viruses enter into the cells. This has all been studied using a super resolution microscopy. So as I was saying, when you combine the advanced resolution from these techniques and viruses become an amazing subject of a study thanks to this advanced resolution that we have, and that has already been taken advantage of, and we have made quite a lot of discoveries in all those fields. And generally, it reveals also the enormous potential. HIV was one of the first viruses that were extensively studied using these techniques, but of course there are other viruses, and this is precisely what is right now done, that these techniques are being applied to a variety of other viruses, well, including SARS-CoV-2. So just to wrap up this theory, I hope I have been clear enough about this, but just to take the take-home message from this is, well, that a fluorescence microscopy is a powerful discovery tool in science. And the second thing that I want you to sort of take away is that we have now techniques which break the super resolution, the resolution of the standard fluorescence microscopy, that the barrier has been broken. And now we can use this fluorescence microscope to study virus structures. And with that, we're gonna do a little bit of practice. And let's take this into practice and let's use the microscope and let's image some of the samples. So just to give you a bit of a background, what are we going to image today? First of all, we are going to image virus molecules, HIV virus molecules. And like I showed you before, this is a diagram of the virus. And just to give you a very basic idea, we're going to study a virus particle, which has two colors. One, which is labeled with green fluorescent protein. This is generally labels proteins that are inside the virus, just to showcase where the individual virus particle is. And we also have a second color. We are going to look at the distribution of surface proteins. This HIV envelope protein is important for the virus entry and infection of the target cell. So these surface proteins on a virus are very, very important part of the virus replication cycle, and we're going to image them. Second thing we're gonna do is we're going to look at the SARS-CoV-2 VLPs. And here I have to tell you something that is very important in science. Scientists try to study things using models. And why is that, especially in viruses? Because of, say, simple safety reasons. As you can imagine, both HIV and SARS-CoV-2 are extremely dangerous if handled improperly. So if you were to study, HIV and definitely SARS-CoV-2, we're talking about full viruses, we would need a much more advanced safety precautions. And unfortunately, I wouldn't be able to be here with you today without all these advanced precautions to study those viruses. We are right now on a campus, we are doing these studies, but for the purposes of this demonstration, I had to use so-called virus-like particles. And while they represent a viruses, they are slightly simplified models of viruses. Think of them as slightly simplified models, but the main important thing is that they're not infectious. So I can actually show them to you. We can work with them rather than working in much more advanced biosafety conditions. So For HIV-1, we have a virus particle that is almost complete. It's just replication incompetent. It's not infectious. But for the SARS-CoV-2, I have something that is a little bit more simple. It is not a full virus. It is a virus particle that simply has a SARS-CoV-2 spike protein on the surface of what is a form of a HIV. And this actually, those models, I mean, why do we use those models? Well, so like I was saying, biosafety concern. Another concern is that if you want to simplify the virus a little bit, we can try to dissect the function of individual components of the virus. And finally, these sort of slightly simplified models are also important when you try to develop vaccines for the viruses. So despite not being full viruses, they are still highly relevant for the studies. So, with that, I am going to stop here just to mention that the SARS-CoV-2 is also, we have a green label inside, and the SARS-CoV-2 spike protein is also labeled in orange. And with that, I am going to stop the presentation, and I am going now to switch the screen to the laser microscope. Okay. So here, hopefully, you can see now the panel, the microscopy, the panel, which is the software of the microscope. Now, first thing we have to do is put the sample on the microscope. So if you look at the camera, I am going to grab the sample. We're going to start with the HIV virus particle. So I have several samples here prepared. And we are going to, first thing actually, if you just so you know, when we study virus particles or study any structures in very, very high resolution, in very, very high magnifications, we have to use oil. Why? Because the objectives that we're using are so-called oil objectives. Normally, objectives are so-called air objectives, which means you do not need any interface between the objective and the sample. But oil objectives require this little bit of an oil because this allows light to propagate a little bit better. So there is in the sample, and so you do not have artifacts, you do not have a light scattering. This is down to the refractive index differences between a glass, air, and oil. So this oil is required for us to get the best quality samples, best quality results. All right, so I'm going now to put the microscope sample on the objective here. Hopefully you can see this as I do. I know the camera is a little bit limited, but hopefully you can still see enough. Okay, so I'm putting the sample, securing it here. All right, that is done. And first thing I'm going to do is I'm going to first look down the ocular to try to focus on the sample, so we are in the plane of focus, and then I will switch to the detectors on the microscope, those detectors, and you'll be able to see it on the screen afterwards. So first I'm going to observe the sample using the ocular. So let's switch here excitation light. You may actually see. And unfortunately, this bit you cannot see, but I see a lot of viruses on the surface. Let me find a good spot where there is not that many virus particles, because there is literally millions and millions of virus particles present on the surface here. Okay, let's look at this area, for example, here. I don't know if it's good, but all right. And now I'm going to switch to the excitation, detection via the detectors in the microscope, and let's start imaging. Yep, I think everything is fine. So let's have a look. I'm not going to go into too much details about the use of the software because that is an entire other lesson altogether. But let's see. Okay, there we go. So as I was saying, see, the image is scanned. As you can see, the image is constantly refreshed by laser scanning across the surface. So what we see here, actually, let me change the focus a little bit and try to explain what we see. So here, those little dots are viruses, and as you can see, there is a lot of them. Green signal is, which is a little bit less visible. Let me make it a little bit better visible. Okay. You can see a bit of an overlap between green and red signal. So these are the virus particles, and they have also a lot of red signal, which is those surface proteins on the surface. What we're going to do is we're going to zoom in a little bit. Let's just choose a small area here, and I'm going to choose an area of 5 by 5 micrometers and we're going to try to now look at the images. Okay, so now we're getting a little bit closer to individual virus particles. Let me just, okay, I'll just switch to the green, okay. Okay, so the green signal that you see is it's individual virus particles. Let me just improve it a little bit so you see it a little bit better. Okay, so each one of those each one of those green blobs is an individual virus particle, and each one of the orange signal, which is quite apparently quite strong, is the signal that is coming from the surface protein of the virus. So let me find an interesting area that we can image this in. Let me go back. I don't think that it looks so interesting, but let me try to find another area. Let me, for example, focus somewhere else. Let's maybe go here. Okay, look, let's just take an image, for example. So, okay, so let's take an image using a standard confocal microscope. So how do a virus particle looks like using a standard microscope? So let's take an image right now. Well, as you can see, you do not see very much. You see some not so bright images. I mean, you see individual virus particles. This, for example, here is most likely individual virus particle. Let me focus a little bit more on them. So they're not. It's a little bit better. But well, you do not have that much information about the position of the orange dye, of the orange color, which is the position of individual surface proteins. And this is exactly what I was talking about. This is the weakness or limitation of the fluorescence microscopy. Anything that is smaller than 250 nanometers will appear as this large blob. And so precisely, those proteins that we have labeled on the surface of virus particles appear as large blobs. But now, let's see what happens when we use the super-resolution microscopy. Let's try to see how it looks like. So I'm going to acquire two images. I'm going to acquire first confocal standard image, and then I'm going to acquire super-resolution image. So let's see what it gives us. Okay. Maybe not so. Let me try again. I think we're a little bit out of focus here, yes. What's happening? But okay, I mean, I know a little bit out of focus, but let's try to already dissect some information of what we see. For example, this over here, we can see actually that the molecule, I say what was before a very large blob, we can see actually that this molecule here, sorry, this virus particle here seems to have only a single, a small amount of envelope proteins on the surface. So once again, let's compare how it looks like in confocal mode. So select here, confocal mode, we see a big blob. And how it looks like in the Stead mode. We can start seeing a little bit better here. I think we are, though, not very well focused on the sample, so let me try to improve it a little bit. I don't think this is such a great place to image. Let me go to an area where there is a lot more virus particles. Let's go somewhere where it's more crowded. See what we get somewhere else. Let's say somewhere here, for example. Oops, that's we're a bit out of focus. I'm now using this panel here. I'm trying to make get a bit more focus. All right. This I think looks a little bit better. Okay, so the green signal once again is the virus particles, individual virus particles, and orange signal is all those envelope proteins that we're detecting. But there, as you can see, it's very diffuse and blobby. So let's try again to image this using superposition microscopy. Let me check if all the parameters are fine, and let's acquire the signal in confocal and in a super resolution mode. Okay, this looks a little bit better. Let me try to improve the quality a little bit. I'll acquire another image. Okay. I think we're starting to see some nice things already. Let's see. I know it's a bit messy, but that's there is a lot of virus particles there on the glass cover slip. So naturally, it is a little bit sometimes difficult to distinguish individual virus particles. But let's have a look. Let me guide you through what I actually can see here. Okay, so well, we have a single virus particle here, I think. What we see, I think, if we zoom in a little bit, I'm gonna choose another option. Let me zoom in here. I think what we have here, if I turn the signal on and off, you can see individual virus particle, and we have the signal, a little bit of a signal here. I think there's a very small cluster of these envelope proteins visible here. If I turn the signal on and off, you can see it a little bit better. Yes, so when you compare it to a confocal image, well, as you can see, well, it's just a gigantic blob. But we see here that those envelope proteins are a little bit on one side of the virus particle. Interestingly, because for example, I think I saw it here. Let's see. This one doesn't. Okay, for example, I think there was another virus particle here. I think maybe there's two of them here. But what you can see here a little bit is, I think what I can see here is a bit of a semicircle here. And the semicircle indicates, well, that there is actually, those proteins are a little bit more spread out all over the virus surface. I think there is a bit of a semicircle here and another small faint signal over here. So this indicates, well, that this virus particle, for example, has surface envelope proteins a bit more different than the other one. This one seems to have them a little bit more spread out. There is, I think, one here and a small semicircle of them here. I think we can see it. Maybe I saw it in another one as well. Let's see what we can find out here. No, not so good here, but let me see if I can find another good example of these differences in another part of the image. Let me zoom in into another area. I'll just arbitrarily choose one. Let's see what we got. Quite a lot of virus particles. No, not so good. Let me try final time, try to find somewhere where there is not so many. Maybe here, let's try here. A little bit messy as well, not so great. I'm really trying to say, well, that is part of a discovery. I mean, you have to find areas of interest yourself. And well, often the samples, will not give you an immediate beautiful result immediately. So let me focus a little bit, see what we can get. Maybe a bit of a better result. Nope, this is not such a great area. Well, what I can do is I can try to go somewhere else and see we can find a nice area of virus particles. I'm moving now the sample around a little bit, and we're going to focus on a different area here. Okay, let me focus a bit. And try here, for example. This looks like it's not super crowded here. Okay, so this is confocal image. This is standard microscopy like before. Let's focus a bit. Individual virus particles. And let's image it in high resolution. Okay, so we're going to acquire first a confocal image and now a super resolved image. Okay, well, once again, I think we have an example of a semicircle here. There is probably one or two virus particles here. And then, okay, actually over here, we have another example of a virus particle, which has only a single molecule, single envelope proteins on the surface. So let's compare those, for example, those two. So how does it look like in confocal? Well, we just get a bit of a blob. And in here, we get this type of a result. And in a semicircle, well, in a confocal, well, we also get a bit of a blob. So as you can imagine, if we study this carefully, superposition microscopy allows us to determine the proportion of virus particles which have only, let's say, single cluster of envelope proteins, or it has a more semicircular, more distributed clustering of the envelopes proteins on the surface. And that to us already actually was an important discovery, because we found out that, to put it into context, we found out that the clustering, the disposition of those envelope proteins on a surface is very important for the virus to be infectious. So those proteins need to be clustered into one small cluster for the virus to be infectious. And so this would not have been possible with standard fluorescence microscopy. Only we were able to discover this using this advanced superposition microscope. So this is in a nutshell. This was in a nutshell looking at how we image virus particles of HIV. Now, let's try the other sample. Let's try the SARS-CoV-2 virus-like particles. Let's see what we get out there. And with SARS-CoV-2, I looked at those images. I looked at that sample a little bit before. And I think, unfortunately, it's a little bit less well labeled, but I think we should still see enough virus particles to try to make a little bit of to get some results out of it. Okay, so once again, I'm gonna put the oil on the surface of the cover slip where the virus particles are. The virus particles obviously are on the inside part of the cover slip. And we're gonna put it here again, like before. Move the stage a little bit. And I'm going to, like before, what I'm going to do is I'm going to first look at it, look at the image using the ocular, just to focus the sample a little bit. Okay. Yep, there are virus particles here. There is definitely less of them. Not as concentrated this sample as the one for HIV, but let me maybe find an area that has a little bit more of those virus particles. Well, let's have a look anyway. All right, I think this was good enough. All right, so let's have a look at I'm gonna just call this sample number 2. That was before imaging sample number 1. Okay, all right, as you can see, the labeling, the number of virus particles, the green one, is very, very sparse. So the number of virus particles that we have is very, very well different to the other sample that I showed you. Okay, so let's focus on one small area. Let's try to find something interesting. All right, I'm gonna try to, for example, I think focus on here. We are going to see, I think, just only some small individual virus particles. Yep. Okay, so yeah, unfortunately, this sample has very, very much less virus on it, but we can still see things, I think. Okay, so let me focus. So yeah, once again, these are individual SARS-CoV-2 virus-like particles. Let me just double check. But what we have, unfortunately, is that most of these virus particles do not have any signal, orange signal associated with them. Some of this orange signal is generally present away from them. That means the labeling is not so great. But let's try to find a virus particle that may have some labeling. All right. I'm going to try to improve the signal a little bit here. And see if we can find something interesting. I am now trying to find the virus particles that may have some signal on top of them. So obviously this sample is well not as well labeled, simply because I think what might be happening is that okay, here we have something. All right, here we have actually something. Okay, so yep, there is a virus particle here on the bottom. There we go. Virus particle here on the bottom, and there is a bit of an orange signal associated with it. There's this large blob. So let's see how it looks like. It looks it's moving a little bit, but let's see what happens when we image it. Okay, I'm going to try superposition microscopy and confocal microscopy. So first image is going to be confocal, and second, let's see how it looks like in super resolution mode. Oops, very, very faint, I think, but let's have a look if we can find any. Oh yeah, well, this actually has, I think, fairly faint signal here. It's very, very diffuse. So it's not the greatest signal, but we see, I think, a small, I would estimate it that there is only 1 or 2 spike proteins on the surface here. Well, this actually tells me something already about the sample in general. I think that this virus-like particles, this preparation of virus-like particles, does not seem to have very many spike molecules on the surface. As you remember from HIV, there was quite a lot of orange signal, HIV virus particles having, I think, quite a lot of envelope proteins. But this is still, well, one of the early models of those SARS-CoV-2 virus-like particles that we're studying, and it's a little bit imperfect. So that indicates that there is, I think, quite a lot of room for improvement in terms of studying those these particular samples. Let me try somewhere else. Maybe I'll find something better. But I already can see that this is, I haven't imaged this sample very much, but I can already see it. So, well, it's not as great as the HIV sample. Let's see, what do I see? I'm trying to find an area where I can see that there is some overlap between the green dots and the red dots. I think there is one here, two of them even. Let's have a look here. Okay, yeah, there we go. Let me focus a little bit and see what we get. So first confocal image, and now let's see how it's, oh yeah, there we go. This looks very interesting, actually. Okay, so, well, while I was saying, unfortunately, it looks like the virus particles have only very, very little virus spike protein on the surface. But this already actually tells me something interesting. We can see, actually, that there is a bit of a separation from the green signal, which is roughly the center of the virus, and the orange signal. That indicates to me that there is something that this spike protein and whatever is labeled is actually very, very large. And maybe if you can remember, in terms of when we look at the HIV, those orange signals were very, very close to the green signal. And so actually we already have some interesting results here, I think. So let me try to image one more time, and then I think I will stop here. Let's see if I can improve the resolution a little bit and image again. No, I don't think that works so well, but yep. Let me go to the gallery and look at that image that we acquired again just before. There it is. So once again, I mean, looking at the confocal resolution, we have a big blob, so we are not exactly sure where this spike is. But if you look at this, we look in the super resolution mode, we can more precisely localize this spike. And as I can imagine, if there was maybe 1, 2, or 3 of those spikes on the surface, in this mode, we would not be able to really see them. You would see very large blobs of orange fluorescence, and only in a super resolution microscope we'll be able to assign where exactly are on the virus surface. So I think what I'll do is I might actually stop here with the acquisition. What I'm going to do is I'm going to open the presentation again, and I'm going to, I think, let's talk a little bit about what we saw here. Let me actually find the presentation, I think. Here. All right, there it is. Okay, so let's go to here. Oops, let me go very, very fast, fast forward this. All right, so let's interpret the results a little bit. So what we saw, what you may have seen. So this is an example from actually the study that I did, very similar to what we imaged with HIV. So in the standard confocal microscopy, we saw those green dots, and we saw these orange blobs. But when we turned on a super resolution microscopy, we started seeing that the green signal is very much localized with either, for example, a single or I think 1 or 2 clusters of envelope proteins. So in respect, the enhanced resolution gave us more information about what is happening in the structure of individual virus particle. So if we look all over here, in confocal mode, we get signal like this. The blue circle is where roughly the virus is. But in a super resolution microscopy, we're able to discriminate exactly where those proteins are. And instead of just a big blob, we can actually assign exactly where on the surface of the virus those proteins are. And that is already quite an important achievement since normally in fluorescence microscopy that was not possible. Now, conversely, this is an example which I grabbed a little bit earlier. When we interpret the results for SARS-CoV-2, you can see once again that in a standard confocal microscopy, you just get orange blobs. In the superposition microscopy, you see a signal here that is much more discrete, and we can see exactly where it is. But another interesting thing I was mentioning earlier, when we look at the distance between the orange signal and the center of the green signal, for HIV, it is very, very close. But for SARS-CoV-2, I can see that it's actually a little bit of a distance. And I think what is happening here is because here we actually have a SARS-CoV-2 spike protein that has on top of it ACE2 protein, which, as you know, is the receptor for the SARS-CoV-2, and only we label the ACE2 protein. So what we have here is a combination of spike protein being very big, and also on top of that there is another protein which we label ACE2. And so this creates a very, very large complex, which is a bit more further away from the virus. So here we can actually differentiate it from how HIV looks like, that there is a bit of a distance. And once again, using a confocal microscopy, you wouldn't be able to see it. All you get is a very diffuse blob, which you cannot really localize very specifically. So already, thanks to, well, this very preliminary study, I already see a little bit more about the virus structure using a very simple, relatively simple fluorescence microscope. All I did was label the samples. All I did was, prepare those slides, and I just put it on a microscope and I started imaging it. And so thanks to advances in fluorescence microscopy, I can very easily get information about structure of the viruses without resorting to such thing as an electron microscopy. So this is exactly where the power of the fluorescence superposition microscopy lies for studies of virus particles. And then, of course, the story doesn't end there, because we can go more. We can then combine not only virus particles, but we can look at them when we study how do they interact with cells. So on top of it, here you have this HIV virus particle as it interacts with a T cell, which is a target cell of HIV. We can see a single virus particle here, and the orange color is the envelope proteins of the virus, and the blue color is the individual CD4 receptors for HIV. So we can actually see how HIV-ENV is interacting with CD4 molecules as the virus enters into a cell. Here we have another example where we did a study where we looked at how dendritic cells, which are important cells of the immune system, are engulfing virus particles of different types. Here we've shown cases that they engulf HIV and Ebola virus particles, and they put them into this specific compartment, which is important for then disseminating those viruses to other cells. So we were able to use advanced superposition microscopy to very precisely get the architecture of this compartment. And of course, what I'm not showing here is then we can extend this further. We can use it for live cell imaging. Imagine looking at those virus particles live as they enter into the cell. All this is possible with this type of a microscopy. Of course, it requires more preparation, more advanced experiments than I'm showing here, but it's definitely possible and indicates how powerful this technique is for study of pathogens, study of viruses, bacteria, anything that is smaller than 100 nanometers. Of course, cells as well. So I am going to actually here finish off, and I would like to first of all acknowledge the group of Javier Martínez-Picado. This is my group at IrsiCaixa for enabling me to conduct my studies here on the campus and study HIV and other viruses. Of course, I need to mention other groups from IrsiCaixa, group of Nuria Izquierdo and Julia Blanco, because they provided me actually for the reagents for SARS-CoV-2 for this preliminary sample that I could put here. Now, finally, what I'd like to do is, if you would like to find out a little bit more about what I talked about here, I recommend you go into one of the reviews which I wrote in 2018. It talks about microscopy, it talks about virus particles. It combines those two quite big areas into a review. So here is the reference and a QR code if you would like. And finally, if you would like this presentation, I provide it in a form of a PDF, and it's available to be downloaded using this QR code. And finally, My YouTube channel has the animation if you'd like to have a look at it again. And also, if you feel free to email me if you have any questions about any of the microscopy, any of those studies that are shown here, or chat a little bit about how to be a scientist, how do we as scientists study this, how do we develop our careers to be here and study those amazing things. And with that, I will stop and I'll thank you very much and take any questions that you may have. Please ask me any questions. You can write them, you can speak them, just raise your hand.
Thank you, Jakub. Terrific presentation. Thank you very much. So this is the opportunity to ask him questions. As you have seen, he is an experienced researcher, and I think he is a great example of a scientific career. So you have this great opportunity to interact directly with Jakub. So any question you want to make, you can do it either online or just write it down on the chat. You can do it in English or Spanish or Catalan, whatever. So if there is any problem with that, we'll translate it into English for Jakub. So please go ahead, and if you have questions, that's the moment.
Okay, let's see.
While the people goes and ask questions to you, Jakub, how relevant you think, Jakub, is going to be microscopy in biological sciences within the next few years? I mean, super resolution microscopy has not been around that long, or not even 10 years now. So what is going to be the role of super resolution microscopy and light microscopy within the science in biomedical research?
Personally, I think it is actually going to gain even more importance over the coming years, because right now we're in about the time where this type of advanced microscopies are sort of starting to come out from becoming more and more popular as they start ending up in many different institutes and they become as a standard. Whereas before, those techniques were kind of exotic and difficult to be able to use them. Right now, it is a time of proliferation of these techniques. And in terms of next several years, I think those techniques will gain a huge prominence, specifically because they're life-cell compatible. Fluorescence microscopy, advanced fluorescence microscopy, if you can combine these advances in the resolution and combine it with life-cell microscopy, is going to be incredibly important to study virus cell interactions or cancer or anything else, but very importantly, that utilizing the advantage of life-cell microscopy. So we can study things as they are, not when we fix them, when we chemically treat them. We can study things in their physiological conditions. So I believe this actually there is a huge field that is already opening up for a lot of studies, and they will bring a lot of new results that were not available before when you combine the advanced resolution and live cell imaging. So I think this field is actually going to grow quite rapidly thanks to all these advances. Because as scientists, we want to study things as they are, not as when they are some sort of a chemically treated models. We want to study things in physiological conditions.
Great. And I don't think we have questions. Is there anyone who wants to jump in and ask any questions to Jakub?
I think there is one hand raised, so I think that the school. I think one of the listeners has a question. Maybe they want to say it. Maybe if we can give them access to speak. Not sure.
Let me see.
Let's see if we can.
It's from Escola El Pilar, from Premià.
Mm-hmm. Yep.
And I'm not sure whether you can directly speak on the mic. And maybe we'll listen to you or not, or maybe we need some help from the organizers.
Let's see.
So he or she is a student from secondary school, third grade, in, I guess, Premià de Mar. Yep. I don't see the second part of the...
Let's see. How can we...
Premià de Mar, yeah.
Yep. maybe best if you can chat, if you can maybe write a question on the chat if possible for you, because I'm not exactly sure if we can right now give rights to the to the person to speak.
So while this attendee is, writing down the question on the chat, I have another question for you, Jakub.
Yep.
So specifically in the field of virus, we've been using for decades electron microscopy. And we thought that was the only means to look at the viral structure. Now we have this technology, super resolution, and on the top of that, we have the ability to look at structural techniques for looking also into the virus. So my point is, because all three techniques, electron microscopy, structural studies, and light microscopy, they would provide, in my mind, complete different information. Are there techniques that are helpful to correlate all those studies, like specifically electron microscopy with super resolution microscopy, maybe also structural techniques?
Definitely. Actually, this is one of the big fields again right now. It's called CLEM, which is called correlative light and electron microscopy, and effectively, precisely, it does what you were saying. What it does, it takes this exactly same image and exactly same part of the cell or part of the virus, and images it first using fluorescence microscope, and then images it using electron microscope, and correlates it with fluorescence microscopy. So effectively, because precisely the results that are often different, well, not so much different. I think fluorescence microscopy has different strengths than electron microscopy. So like I was saying, with fluorescence microscopy, we can visualize specific structures a little bit better, but electron microscopy has even better resolution. So when you combine those two, when we combine those two, you actually can get even more information, and that is quite a complex field of study, because as you can imagine, combining this type of microscopes with electron microscopy requires either very, very delicate samples handling, because you have to know exactly where have you been. You have to then put it in a different instrument and then find exactly the same area. But it is being done, and there is a lot of studies right now, actually quite prominent and amazing studies, which utilize this type of approaches. Not in every laboratory it's possible, but it's a definitely exciting field of science to get into it.
Super. So we have the question from Cinta Lamagne, and she is asking, do you think COVID-19 vaccine will be effective?
Oof, that is a very, very broad question. I mean, I am a virologist, but immunology and vaccinology. Well, look, all I can say right now is the studies, the studies that are coming out from the vaccines that are right now available look promising. I think, as you can imagine, there has been a lot of effort put into getting these vaccines, and there is a lot of greatest minds, greatest scientists, which are working right now on those vaccines. So I think what we're experiencing right now is unprecedented amount of resources that are being used for the study, and how quickly it was possible to get these vaccine candidates is actually amazing. So I think, given actually from my personal opinion, given that this virus is relatively simple compared to, for example, let's say HIV in terms of its replication cycle. And well, this is just my opinion, but I think the finding of the vaccine, unlike finding an effective vaccine for HIV, will be simpler. So there is a good chance that these vaccines that are those candidates will actually be effective. So I'm quite positive about this.
I have to say, to add on the top of that, that I completely agree with Jakub. Despite of the pandemics, SARS-CoV-2 is a much simpler virus than other viruses. If you compare that with HIV, completely different, and different levels of latency, problematic. So I completely agree. So there is another question from the Escola del Pilar in Premià de Mar. And the asking, first, they thank you for the presentation. And their question is whether, what happens if HIV contacts with another virus, for instance, SARS-CoV-2. So let's say that the two viruses infect simultaneously one cell. I would understand that.
Yep.
How do they compete each other? What is your estimation?
Okay. Well, fortunately for all of us, these viruses are from very, very different families. So actually, they utilize very different mechanisms for replication. So actually, even if it would be possible for those viruses to infect the same cell, which is not, because first of all, HIV infects the cells of the immune system, or SARS-CoV-2, we're talking about the epithelial cells. Even if they would be able to get into the same cell, they would most likely do not interact with one another at all, because they utilize different proteins, different mechanisms, and so there is no danger ever of having some sort of a strange, horrible hybrid. Those things are a bit more. This is actually a bit of a science fiction, especially if you study structures of the virus particles in terms of how they replicate, what sort of mechanisms they utilize in cells, and they are largely independent of one another. So really, there is no chance of them ever mixing together on a cell level. And even if they do, even if somebody stick them artificially together into a single same cell, they would most likely, they will not interact with one another. So I wouldn't worry about that.
Yeah.
What do you mean, like mixed together? I'm not quite sure. As in, you mean to combine, they basically become one? No, viruses cannot well infect one another. If you think about it, viruses actually require cells to be infected, because well, we compare a cell size to a virus size. Virus needs if we look at things from I guess a point of view of the virus, a cell is like a big field of flat or undulating field, and it needs that sort of a flat membrane to infect. If the virus would encounter another virus, they do not have appropriate receptors, they do not have appropriate structures to be able to merge and mix into another. So no, that is actually physically impossible due to the physics that govern this type of reactions.
Perfect. Another question from Marina Cunquero.
Sure. Okay, let's take it in. Yeah.
I think that the question to the people at the Escola del Pilar has been clear. I mean, I think that's this point. So Marina Cunquero, she is asking. She's again also thanking you for the presentation.
Oh, great, happy to help.
The demonstration of the system capabilities. And she's asking whether it would be possible to program the microscope so it acquires multiple samples automatically, like kind of high throughput for many sample acquisition.
Oh, absolutely. I mean, imagine this microscope over here, and there are special add-ons to this type of a microscope, and they're called sample hotels, where basically you put in a lot of samples, and then the microscope, or you can use imagine a robotic arm or a slide sort of a holder, which then puts the samples in, images them, then grabs another sample and then puts the next one. Definitely, microscopes can be used for high throughput studying. This microscope is more of a research microscope, so we do not have this kind of capability. But it's actually many companies offer this sort of microscopes. They are often used by pharmaceutical companies which want to look at a lot of different drugs, for example, and scan those many microscopy samples treated with different types of drugs and image those samples. So yes, that is quite possible.
Consol says, congratulations, very interesting presentation. Thank you, Jakub, and thanks to UVic-UCCAdreçat too.
Well, no problem. Happy to demonstrate. I hope it also brings shows you how scientists work, how we actually work, how exciting this field is, how many interesting discoveries you can make, and how cool the microscopy is actually. So yeah.
I just want to point out that this coming week on December the 1st is the AIDS Day. And I just want to mention that because the research that has been done in HIV has been very useful for the quick advances in the COVID-19 research. So it's a great example of how science is developing one field, And all of a sudden, when the need is there, it can automatically and quickly jump into a different field, in this case from HIV to SARS-CoV-2, and make a very quick advance in science. So I just want to say that. I wanted to say that because these days there are many journalists who are approaching us to, compare HIV with SARS-CoV-2, and I think it's fair to say that many years, more than 40 years actually of research in HIV has led to quick advances in this pandemic for SARS-CoV-2. And I think Cinta Alemany has another question. How much does your microscope cost? I want to buy one for my high school.
Okay, one for high school. Basic microscope will, unfortunately, of this quality will set you around 300,000 euros. More advanced microscopes, we're talking about €500,000 up to €1 million. But if you want to get a fairly basic microscope, I really recommend you look at those Lego projects, because you can build a competitive microscope of 1,000 or 1,000 or 2,000 euros. And of course, already more research microscopes will set you, a few thousand euros. It's related to complexity. But this type of a microscope, we're talking about 300,000 euros, unfortunately. So yeah, look at those Lego microscopes, and those actually can be built and can be quite useful.
Perfect. Another acknowledgement from Maria Eugenia Bilowi for the presentation, Jakub.
Right.
Okay, so I think that this is all. If there are no further questions, thank you, Jakub, and thank you to the organizers in UVic-Divulgar for this wonderful seminar, and we'll meet in a different occasion with all of you. Thank you.
Definitely. Thank you very much, and everyone take care.
Bye.